Due to their significant potential in regenerative medicine, increasing attention is being focused on the biology and manipulation of human embryonic stem cells. In this video, we show you how to grow and maintain human embryonic stem cells in a pluripotent state using mouse embryonic fibroblast feeder cells in feeder-free conditions on Matrigel.
Hi, I'm Zhang from the laboratory of Michael Teitell in the Department of Pathology and Laboratory Medicine at the University of California, Los Angeles.
Today, we will show you a procedure for culturing human embryonic stem cells on mouse embryonic fibroblast feeder cells and in feeder-free conditions on Matrigel. This procedure involves preparing mouse embryonic fibroblast cells, generating MEF conditioned media for growth on Matrigel, preparation of Matrigel plates, and methods for feeding and growing human embryonic stem cells over time.
So let's get started.
To begin the culture procedure, human embryonic stem cells are plated on mouse embryonic fibroblasts, or MEFs. To split human embryonic stem cells plated on MEFs, we start with confluent human embryonic stem cells and plate at a dilution of 1:6 to 1:10. Depending on the particular embryonic stem cell line, this will become confluent again in 5 to 7 days after splitting.